Afternoon, everyone. Okay, so today we have Professor Euiheon Chung with us for our colloquium. And so unfortunately, so he's very, very busy. So unfortunately, he couldn't come in person today. So hopefully we can have him in person next time. So hope we have a good chance. And so I'll briefly introduce Professor Chung. So he graduated from KAIST and he obtained his BS and bachelor's degree and also his master's at KAIST. And then he moved on to MIT for his PhD. And at MIT, he joined the Harvard MIT Health Science and Technology PhD program. So this is a very unique program where you get interactions between like clinical studies and also engineering. So I think you guys are aware of BME starting or UNIST starting undergraduate courses for Ulsan Medical School students, right? So that is actually designed to be hopefully better than this program that MIT and Harvard has. And so Professor Chung is very, very talented in engineering and also very, very translational studies. And so he has received many, many awards, but I don't think I want to spend too much time for the introduction. So, and after his PhD, he also did his postdoc at Massachusetts General Hospital and Harvard Medical School. So today he's also going to talk to us about how we can use optics for translational studies in the brain. So please give a hand for Professor Chung. And without further ado, I'll pass on the mic.
Okay, thank you, Professor Park Jung-hoon. Thank you for overly good introduction for me. So today, I'm very happy to meet with you guys in BME department in UNIST. So I will start with my talk with a title as a Translational Neurophotonics. And a subtitle I put as an imaging and manipulating brain diseases with light. So first, what is translational neurophotonics? So we first talk about neurophotonics, and neurophotonics can be considered as a branch of biophotonics. And you can also see biophotonics is a combination of bio and photonics, which involves optical imaging of target molecules, cells, or tissues, as well as optical monitoring of interventions to disease progression. So when we talk about translational neurophotonics, which is putting translational so that our application is translate from the basic science of neurophotonics into the clinical trial or applications. So which we talk about, for example, in here, like brain tumor or many other types of neurological diseases, which poses a formidable challenge for us. So for this, we do need a transdisciplinary approaches at the interface of neuroscience and biophotonics. So we want to diagnose and guide therapeutic strategies for clinical neurological disorder. So first, let me discuss about the imaging aspects. Obviously, our target for this neurophotonics research is involved in the brain and nervous system. And you can consider many orders of magnitude are ahead of us. For example, you can consider tumors growing in the brain. You can observe it in a larger scale, we call macroscopic, and down to the nanoscopic. For example, you can try to observe blood vessels inside the brain, which is about microscopic scale if you are looking at the capillaries. And even down, further down, you can consider neuronal synapses, which has a connection between neurons to another. And those synapses sizes are very tiny. It's like 1 micron or below. So we do need a higher resolution technology, which we can call nanoscopy these days. And this covers that how much of the multiple dimensions we are dealing with. And obviously, when we want to have a high resolution, we have to sacrifice penetration depth. So there is some kind of trade-off. First of all, I want to start with a movie which I took. Here, the green color is a tumor cells inside the brain. And you can see this video ray to photomicroscope to see this dark dots, which is a RBCs, which absorbs the light. So you can see the actual RBCs moving inside. Even with about the same blood vessel size, you can see which here the cells, the perfusion of blood is not very well. You can see intermittent, like a little bit hinder the blood flows. So this actually shows that is a blood flow is a very important, and you can use a functional microscopic imaging to tackle this problem. For example, you kind of know about like the vascular issues which feeds the brain tumor, and also very important disease such as a stroke is involved with the blockage of the blood vessel. So in this case, the biomarker is a measurable indicator of some biological process or condition, and we can consider this blood flow as a biomarker for healthy and diseased brain. So I'm going to discuss about how optical methods can help for measuring cerebral blood flow. You may have heard about Doppler, so laser Doppler flowmetry shines a laser light and then detects the changes of a back reflected signal to figure out the bulk tissue perfusion. Unfortunately, this is only providing relative measure. Also, there's a technique called the optical coherence tomography, or OCT, and there's also Doppler OCT method, which also gives some quantitative measure of cerebral blood flow. Today, I'm going to show you RBC tracking methods, which uses intravital microscopy to see a single vessel and single cellular level to track and be able to quantify the blood flow. And the other technique I want to discuss is a so-called laser speckle imaging, and which I'll discuss a little bit, but it shows a perfusing of blood flow with this contrast. So I will start first with the laser speckle imaging, and then second, I will talk about RBC tracking method with a advanced microscopy techniques. So this figure, you may have, everyone probably have used a laser pointer, and when you shine laser pointer to a surface, which has like at least, even if we were polished, there's a several micron changes. So the laser light can create this very high contrast, granular, almost noise-like pattern, and that's called laser speckle. If you look at closely, it looks like a particle here. So let me introduce you a technique for functional imaging by use of laser speckle. And what is laser speckle? And that laser speckle is when you shine laser beam onto a surface, that microscopic reflections and interference creates this random looking speckle pattern, which normally we don't like it because it's noisy. For example, when you are having an ultrasonic image for a mom watching the baby inside, people usually try to remove this speckle because it actually is interfering. However, we can utilize this speckle for some of the purpose, for example, like blood flow imaging. So this figure probably gives you an idea that during the time of a camera exposure, when this lady, which is stands still, has a very high contrast images, while the underlying subway, which is moving, you can see a blurring effect. So actually, when you are trying to analyze how much blurring effect is coming, then you can see there's underlying movement and dynamics there. So that dynamic information can be encoded into the speckle. For example, in this picture, this is a white light image. Just take a picture. You can see blood vessels a little darker due to the red blood cell absorbance of light. However, by use of this speckle contrast, just seeing a relative, standard deviation of the speckle, you can actually achieve this very strong high contrast images. And this high contrast actually reveals the underlying scatter, movement, and dynamics. So I start to use this technique for studying stroke. So stroke, especially ischemic stroke, means a stroke caused by a blockage of blood vessel. So there's multiple ways of generating stroke, but I used so-called photothrombosis. So this uses a dye called rose bengal, which is a light sensitive. Once you give a green light into this, then it will cause a blood blockage, occlusion, so that you can cause photothrombosis. So I made a small gadget which focuses a green light onto a mouse brain, and you can actually create a single focal ischemic stroke with this device. So here's an example of experimental scheme for speckle imaging with a focal photothrombosis model. So you can have this rose bengal can be injected IV, but it's only after 1.5 minutes, you have to shine with a green light like this. Here I'm shining at the middle of the blood vessel, and then afterwards, you can actually observe. So what you see though, this row is a white light image. So even after this photothrombosis, you see the blood vessel still there. However, when you are trying to look at with a functional vessel by use of speckle imaging, you can see the vessels here is gone. So because there's no flow anymore. So you can actually use this one to observe a little longer time points. For example, every 5 minutes, we actually observe this, then you will see the blood flow is slowly coming back. So this phenomenon is called a reperfusion, and this is known that this reperfusion can be clinically important because when some people got a stroke and then recover, but this abrupt reperfusion may cause a following problem, which is important clinically. So now I actually use this functional brain imaging scheme with the electrical sensory stimulator to give an electrical current to the hind limb of the mouse brain. And for this green light duration, I give an electrical stimulation and tested whether laser speckle contrast can be useful for this functional brain activation. So what this gives is the relative contrast of speckle actually shows up at this location, and so that I can actually show that my student RHO actually performed this experiment. So for this 5 seconds of stimulation, you can actually see that the speckle actually contrast is changing. And at the same time, you can perform another modality of optical intrinsic signal to see that this is actually happening reliably. So that this gives us an example of electrical stimulation can be useful for, you can see the functional changes by use of laser speckle imaging. So here, so far when I discuss about laser speckle imaging, in fact, I never talk about any quantitative manner because the limitation of the current laser speckle contrast imaging is actually semi-quantitative. So here I was thinking about how to make this to be more useful. For example, when you develop a drug which involves modulate blood flow, then unfortunately you cannot easily test the drug with the current laser speckle imaging because it's not absolutely quantitative. So we were considering how to make a quantitative approach by use of this. So here comes my background of aerospace engineering. I did my master in combustion engineering, and there is a technique called a PIV, or called a particle image velocimetry, which is you want to see the profile of flame. You actually send out a particle and take a very fast double shot image. Then you can analyze this to find out the velocity field. So I consider that laser speckle almost looks like a particle. So even if it's not a real particle, can we consider this as a particle and adapt the method of PIV or particle image velocimetry here? So for example, this is a first shot of laser spectral image, and this is after delta t, the second shot. Then you can actually make a small window and then try to correlate these to find out how much of average displacement has happened. So that, for example, you can actually use this one, analyze it in Fourier domain, and then you can back Fourier transform to get cross correlation. You can do this multiple times, and this is an average of cross correlation map, which provides in space domain, you can see the average displacement. And by knowing the delta t and this average displacement, you can actually draw a one vector, which represents this area's velocity vector. You can do this analysis for over this region, and you can create velocity map. So that's a very simple idea, and we test this one and named it as an optical speckle image velocimetry. How to realize this? And the difference from this approach from the previous one, we depend on the smearing of laser speckles. But for this particle image velocimeter, we need a very sharp images of speckle. So we need a very sharp and very short exposure time, which actually is below the camera's minimum exposure time. So we had to use so-called another technique for the AOM or acoustic optic modulator, which allows us to control the beams in even microsecond scale. In this case, we use just 1 millisecond scale on. Even if the camera exposure is on, we only give light at this instantaneous time so that we can have a very sharp speckle at each time. So by this, we can have this first speckle light, and then you can take images of a very short delta t to correlate a consecutive speckle images. So for testing this, you need to have a simpler model. For example, translational stage, you can have a true value and you can correlate. And also you can use a fluid phantom experiment with a known flow velocity, and then you can correlate. So, and finally, you can try in vivo experiment. For example, we first conducted a calibration experiment with a translational speed versus this optical speckle image velocimetry, which correlates very well up until about 7 millimeters per second speed. So with this, how to test and show the importance of this technique. So we also used a stroke. So the pre, during, and post-stroke, we actually did the experiment for this. So this movie shows a speckle images and the analysis here. So that small little square, I actually show this analysis of velocity vectors. And also you can calculate the actual speed, which is shown on this downside. What's notable here is now you can see this color scale bar. Now it has speed millimeter per second. So now we have a unit which is quantitative and velocity and speed. So for example, to show how dynamic this phenomenon is, we chose 1 point P over here, and every 26 milliseconds we plotted the velocity vector before, during, and after. You can actually realize how dynamic the speed, the direction, and the amplitude of the velocity vector. So this speckle velocimetry are demonstrating a 2D quantitative maps to reveal this dynamic nature of blood flow velocity maps during the process of vascular occlusion. So now let me change the gears a little bit from the speckle, and the issue of the previous presentation is limited by two-dimensional space of flow. So we have been considering the actual blood flow inside the brain is actually three-dimensional. So how can we go deeper than the surface flow? So here comes our idea that axial scanning by scan confocal microscopy with ETL, or ETL stands for electrotunable lens. So what is ETL? This is a lens but by use of electrical activation, the focal length of ETL can change. What that means is without physically moving this lens up and down, you can actually control the focus of this lens electrically and relatively fast. So as a basic study, we actually used this ETL or electrotunable lens by driving a current. So for example, when you drive current, the focal length changes. However, the problem of typical ETL has very short ranges. And our idea is what we want to have is almost a volumetric imaging, which requires the axial focal length changes as long as possible. For that, we actually run ETL at its driving frequency at resonant mode. So when we find out resonant mode, such as 400 Hertz, the axial range now become approaching to about 100 microns, which you guys probably know that confocal microscopy will about looking at down about 100 to 200 micron depth. So this is kind of perfect for us. So by use of this, we create a microscopy with a confocal axial scanning, and this is the so-called point spread function. For those who are not familiar with microscopy, this point spread function means a simple point shows up as an image here. But this is a three-dimensional, so this is a lateral point, but axially we want a point spread function to be elongated to cover all the depth. So by use of this ETL at resonance mode, we actually cover very long depth of this axial effective point spread function. And by using this one, you can sweep the field of view. You can achieve very high frame rate. So here's results of axial scanning, line scan confocal microscopy. So for example, this is one single shot, and this is our ETL microscope. So this is a 3D schematic, and this is the mouse brain, which we took one shot of the image. So NZ equals 1. You cannot see some vessels are clear, but the other vessels are not clear. And this one, by conventional confocal microscope, you can actually move ups and downs. You can see some blurring, but at least the focused ones you can actually see so that you can create three-dimensional. However, one single shot, this current one is even without changing, you can see the whole G information as a projection images simultaneously without moving ups and down. So this is about 20 Hertz we can achieve. And you can see the comparison of this one, which at this point, this image actually shows the detail, but the one-shot image do not show that, revealing that the volumetric projection imaging capability of axial scanning, line scan confocal microscope. So from there, the issue is 20 Hertz is like a video rate, but some vascular events require even faster imaging speed. So we are considering how to improve this one. So the outcome of our idea is it would be better to axially scan even more rapidly and axially descan. This is a little bit confusing concept, which I will try to explain now and in confocal microscopy. So what it means is this blue rays getting in. And by the way, this is a so-called tag lens, which is similar to variable focusing lens. And you can see the focuses are changing, and that results in the sample side, the focuses are sweeping. And this way, when you change the focus from here, when you go back through this during the time of the lens, it actually recovers the original path. And you can use line scan confocal microscope to collect all this actual information in one shot. So this is the three-dimensional schematic of the microscope, and it's actually similar to previous one. However, the changes are here, tag lens. So this is the schematic of tag lens, or tag stands for tunable acousto-optic gradient index. What it means is it's a very similar to AOM in cylindrical shape, that by electrically modulating, you can create this kind of a lens, which you can change the focal length very rapidly. In our case, we actually used a 70 kilohertz of this tech lens, which is a super fast changing. And with this, we now can achieve like 10 times even faster rate of 200 hertz for the in vivo volumetric images. So for example, so the left part is like conventional confocal images going up and down. The right side is tech rapid axial scanned and descanned line scan confocal microscopic image. Let's take a look. So let me show one more time. So left is an ups and down, right side is using this one to actually see the projection images of the blood flow. Let me show a little bit more analysis for this. It's inside the mouse brain, you actually can have one single stacked image, which is a color coded here showing 1 to 100 micron depth. And when we are doing this Rassad imaging, you actually collect all the information. However, thinking about the actual geometry of a blood vessel, which is three-dimensional, and what we are seeing is only projection images. So we do need the actual geometry, three-dimensional geometry at least once. So we take one image of stacked image, and then we take this. So what that means is what we are actually collecting velocity is actually projection velocity rather than the actual. So you just need to divide by cosine phi to achieve the real velocity. How to achieve this real velocity is you actually can sweep this through, then you can create this very interesting pattern, which one dimension is a space and the other dimension is time. And you know the slope of delta x over delta t stands for the speed. So with that, we actually can have multiple points, actually many more points to analyze in this data set. We show that depending on the blood vessel diameter, the mean velocities are actually well correlated. Also, we also have to show that the projection image and the actual image can be quite different. Here in relative changes can be 30%. So another interesting aspect of the analysis here is this multiple point. We actually chose arteries and also 2 capillaries. Then you can actually create this type of images, which this axis is time, this axis is space, so that you can have this slope gives actual velocity. What's interesting here, let's take a video here. So this is the actual Rassad image. At 200 hertz, you can see a very dynamic changes. Some things are flowing, usually that's a RBC streaks lines. And you can see veins has a very low amplitude and capillaries a very slow speed. However, arteries can be very high speed over here. Also, what's interesting here is you can actually do a frequency analysis that some capillaries here, this is directly connected to an artery, which also shows this 2.4 hertz of fluctuations in capillaries. So our capillaries, which is close to artery, even can actually reveal this fluctuations which follows this arterial pulsation. So this is only possible by Rassad imaging, and that's fast enough for this hemodynamic analysis. Now, I want to change my gears a little bit from the imaging aspects towards light-based therapy and neuromodulation aspects. Obviously, the issue of the light is so far we have used it for more like imaging or collecting information. But on the other hand, photons also has a energy, and we may be able to utilize this photon energy for therapeutic applications. So the first example I want to bring up is a so-called phenomenon called the Microvascular Store. This in brain and clinical implications. What is capillary store? And capillary store is a brief stop of a RBC movement. And this is believed to be important, for example, like Alzheimer's disease, because in AD patients, their cortical blood flow is dropped, reduced by 25%. And in animal model, what we've seen is 0.5% of all the capillaries are temporarily blocked and then recover. So this phenomenon is called microvascular store. And here's the diagram of the vessels structures from the zeroth order to first, second, third. And you actually can see some of these green labeled ones, so-called parasites. And this parasite, there's a big debate for whether parasites is controlling this capillary flow at this down single capillary level or not. So we are interested in tackling this so that by use of photostimulation for optogenetic stimulation, we may be able to control a single capillaries. For that, optogenetic stimulation unit has been added. And this is just one example of the setup. And I want to show reversible control of microvascular flow by use of optogenetic stimulation of this parasite. Peri means surrounding. So here you can see a red one's a blood vessel and surrounding green labeled ones are parasite. I actually focus on this one vessel. And then when I give a shot, I try to see the blood flow changes. So you see this one spark that gives a one single shot over here. And this black dot stands for RBCs. They are moving because of the flow. But after this shot, you can actually see they start to be very much slow and stop. You can see single RBCs there. And what's interesting is with this condition, they actually recover reversibly. So what this shows that we may be able to having a tool for reversible capillary storing at the single pulse stimulation onto a parasite. So for this, we may be able to study a capillary network because inside the brain, with this limited resource of blood flow, there's a mechanism which we still yet known very clearly at down to the capillary level, whether there requires a neural activity, which may need a more blood flow to that direction. So here is an example of blood vessel network. And here that star mark and that parasite, I actually gave a optogenetic stimulation at 50% of a duty cycle until this capillary is squeezed and then stop. And then we remove the stimulation and see how it works. So here's a movie showing that very interesting shape of capillaries. And I give this part, like this bright spot is actually sending the light photostimulation over here over time. And then you can start to see, so this squeezes and the RBCs cannot move along this capillary network, while the other part of the capillary still moves. And when you wait a little bit more, actually this part will recover spontaneously. And I think I'll show you here a little more. So if you're watching this, it's quite interesting. You see a single RBCs and they start moving. And what's interesting here is because it's a laser scanning, even single shot contains the information of the flow direction and even RBC speed. So now I want to move on to show another interesting topic, whether when we use this light to be able to use as a pacemaker. I guess you have learned what pacemaker is for to make the patient who have a irregular heart rhythm can recover their normal heart rhythm. So in this case, I have a undergraduate students and who actually came to study with me, and she has a special skill for be able to making this egg and dissect this egg embryo and then study this. So with this technique, we actually used this egg embryo, which contains a living heart. And we are trying to put this optical fiber to giving infrared light to be able to see if we can control the heart rate. So here's the movie. And this is basal heartbeat. You can see this egg, small chick heart is moving. And when we give a laser irradiation here, you can see it moves faster, right? So we give a laser irradiation first and then we can go back, so it recovers the original slow speed. So this is an optical pacemaking, and this is the setup, which is quite simple. And you can have this embryo and optical fiber on top, and you can study with varying power, pulse durations, pulse size, and radiant exposure, so that the heart rate with this irradiation, you can actually control the heart rate, and you can study this. So this was a quite interesting study. We actually showed optical pacing can be possible with this thulium laser irradiation, which has a 1927 nanometer. So this undergraduate student is now a PhD student at Duke University. So for this aspect, so we may want to use a photostimulation even for neuronal regeneration. So that original idea is coming from the discussion with the ophthalmology doctors that after LASIK surgery, many patients suffer from dry eye disease because during the surgery, you cut off the terminal part of sensory nerves. And if they don't recover properly, the cornea starts to suffer from dry eye disease. So we were wondering whether maybe this optical technique may be able to help for recovering this. So you can consider in vitro and in vivo model and test neural regeneration, and by use of functional imaging, you can study this neural regeneration based on photostimulation. So we started with the in vitro model first. So photobiomodulation, so-called by use of near-infrared light, we may be able to boost the neurons arms, their growth. And especially we were interested in this cornea, which is connected to trigeminal ganglion. So we actually have a model for mouse trigeminal ganglion. We took out the trigeminal nerve ganglion to making as a primary cell culture. So with that, we want to shine them with the near-infrared light, in this case, 808 nanometer near-infrared light. And we tested two different cases, for example, continuous wave or pulsed wave. So this is a representative one for the neuron with a non-irradiated control. This is cell body, this is axons and dendrites, and this is after irradiation. How to analyze this quantitatively? There's a known tool called a Shore analysis. We want to see how much of a length range do they cover and how many of the branches. So this analysis allows us to quantitatively compare this neurite growth of trigeminal ganglion neuron and depending on energy density and frequency. So first modulation, we were interested whether which frequency is the optimal. So this is the anatomy of this corneal nerve, trigeminal nerve. We actually took out this one to create this 1 day and 2 day with a control and 2 joule of continuous and 2 joule of pulsed wave. So a little bit of this slide, but I think you can see that with different conditions, the average neurite length per neuron and number of intersection and enclosing the radius has been compared. And what you can see is with the different conditions, you can find out continuous and pulsed mode at 10 Hertz are actually pretty good in terms of average neurite length. And also, the other part is we compare all the other parameters. And we concluded that 10 Hertz was the optimal. And at the same time, the energy density is not always good for when you increase. In fact, when you increase, there's an optimal energy density. When you are giving too much, it actually has a detrimental effect. So we found at 1 and 2 joule per square centimeter, generating a best neurite growth. So this is another example of the power of a near-infrared light for neuronal growth. However, this is tested in the in vitro. So we may wonder whether is it also happening in the more complicated in vivo case. So for that, we actually brought a model system to see photobiomodulation can consider as a real therapy. So in this case, we consider which part of the brain organ nervous system would be the most easy to translate to human. And we focus on this olfactory bulb, because in the mouse, olfactory bulb is actually very close to the top surface, which is reachable from outside. While in human, it is actually a very low down of the skull, but that part is may be able to reachable through the nasal cavity. So we consider this as a gray part, and make a disease model. We use a kind of a stroke model, mimicking vascular dementia at the olfactory bulb. So in this case, we made this a mouse model by use of photothrombosis right onto the olfactory bulb. Then the animal will lose their ability to smell. So that's we call olfactory function. And how to test the olfactory function? There's a simple test. You randomly hide your odorous food pellet somewhere, and then after 1 day of starvation, you put the mouse and then measure the time for finding this food. That we call latency time. And when the animal is poor in their olfactory function, it takes longer time. And our question is, we wonder if after this event, we may be able to use a photobiomodulation as a therapy. So we give 2 minutes per day for every day, day 2 to day 7, and then we sacrifice at day 8. So let's see what the results looks like. So first of all, we want to do always control test. So we wonder if photobiomodulation may be able to boost their olfactory function. Remember, the y-axis is the time to find food. So the longer, the poorer the olfactory function. And throughout this event, with giving PBM to normal mouse, it doesn't improve for the normal mouse. However, when we give this one as a photothrombosis, this is a day 3 and this is day 7. And you can find that the photothrombosis, the time for finding food is significantly longer. And even 1 time of photobiomodulation, it actually dropped down significantly. And over time, you can find out after 6 times of photobiomodulation therapy, it very significantly drops the time for the finding the food or olfactory function has been recovered. So this is an interesting kind of phenotype finding for us. And then the question is, what makes this possible? So for that, we start to analyze the olfactory bulb damage by taking out the olfactory bulb and finding out the damaged area. And we look at this olfactory bulb and damage area fraction. So we compare the baseline photothrombosis versus treatment effect by comparing those fraction of damage. And we realize maybe there's some trend, but it doesn't really significantly is different. So we want to plot this to analyze the fraction of damage area versus relative olfactory function, so relative latency time. So the more damage obviously may take a longer time, which is kind of shown in PT, but with the PT plus photobiomodulation therapy. What's a little interesting here is the fraction of damage area and the olfactory function doesn't change. So this kind of appears to be quite interesting, and you want to look at a little more deeper inside. So another aspect is we took out the tissue and compare this a cytokine assay. By this cytokine assay, we actually find out that pro-inflammatory cytokines are increased in PT, but with PBM, it drops down, while anti-inflammatory cytokines are somehow increased in PBM therapy. So this gives one hint for us. Currently, we are using a bulk RNA sequencing to figure out at the transcriptome level what's actually changing. Also, the tissue level immunohistochemistry gives us a tool to understand what's happening, not only in neuron, but also glial cells, such as astrocytes and microglia And also we want to look at the blood vessel changes, and that's indicative of angiogenesis as well. So there are a little bit more works remain to be done, but this hopefully gives you an idea, some example of how light can be useful as a therapeutically. So as a summary, I want to conclude my talk about translational neurophotonics. First, we need to find out problems, which is in our case, preclinical animal models of disease for this unmet clinical needs, such as stroke, and now we are interested in chronic pain problem as well. And the technology, for example, like in vivo optical imaging, probably gives us a quantitative tool, and that makes it possible for our finding to be more translatable. So for this neuroimaging application of biophotonics, I hope I can convince you that this is very interesting for studying neurovascular diseases, and the power of photomodulation may guide us therapeutic strategies towards clinical translation. With this, I want to conclude my talk. Thank you so much for your attention. I would welcome your any questions.
Okay. Thank you so much for the very, very interesting talk. So now we are open for questions from the audience. So you can either use you can type the chatting or just turn on your mic and speak freely. Okay, Euiheon Chung has a question, okay.
Thank you for sharing the good research. I would like to ask if there are studies on the relation between the capillary bloodstream and amyloid beta accumulation. I know some disease named cerebral amyloid angiopathy. It looks like that have a very important connection.
Oh, yeah, Yeon asked a very important question. In fact, that's also our interest, current interest as well. So amyloid beta causes a problem in vessels. So that's called the CAA, or cerebral amyloid, CAA, cerebral amyloid. Sorry. So it gives a problem in vessels. So that's actually known clinically very important. But on the other hand, the real issue is currently with the lack of a treatment for Alzheimer's disease, but it's known that more than 50% of patients from the autopsy, when we took out their brains, more than half of them has a vascular lesions. So we know that there's a high correlation of vascular issue for the AD. The real question is, we don't know whether vascular problem accelerates to the Alzheimer's phenotype, or Alzheimer's phenotype, such as amyloid beta, actually makes the vessel more vulnerable for this lesioning. So this is like chicken and egg. I heard, at least I asked a big guy in this field, they actually do not know. So I think clinical perspective, this is very, very important aspect. As your question, capillary is the most important business end, and the function of capillary might be compromised by amyloid beta. I think this is a very interesting research topic. Thank you.
Thank you.
Okay, thank you very much. Okay, so any other questions, comments? Okay, I actually have many questions, but I would like to ask them later. So the students to ask more questions.
Students can ask in Korean.
Yeah, in Korean. 한국말로 해도 되거든요. 네, 질문 자유롭게 하면 좋을 것 같고요. So maybe I'll just ask a simple question. So one question was, so in the pacemaker work, that was very interesting. It was very cool to me. So I was wondering why you used the 1927 nanometers.
Yeah, a very good question. In fact, that optical pacemaker project was a kind of we made it by law. It's known that this phenomenon has been published in Nature Photonics some years ago with different laser wavelength. We happen to have this a thulium laser that is being used in the 비뇨기과, urology, for the surgery of the prostate, prostate surgery. So we know that this, when the wavelength is longer, like IR range, it has a higher water absorption. So I think this one gives a lot more like heat deposition, and then may convert it into this excitation of this myocyte, cardiac myocyte. And that has been kind of by chance. And at the same time, probably you have heard the field of infrared neural stimulation, where something like this 2-micron laser, when shined to such a sciatic nerve of the mouse leg, it actually gives a optical neuromodulation or neural stimulation. So that has been known. So we actually use that a little bit of a known knowledge. Thank you for asking.
Thank you very much. So I think that's interesting. So if it's due to heat, I think you can also, the options for wavelengths also, there's a lot more options. So okay, thank you very much. Mingyu, okay, so Mingyu has a question.
Yeah,
so
I can see that.
Yeah.
Oh, so I suggest you guys should write your question, any questions to chatting, which will be maybe easier. So Mingyu, have you tried to observe any biomolecular exchange and tissue blood vessel barrier or blood-brain barrier? Okay, very interesting question. I think Mingyu understands the importance of blood-brain barrier, which is a mechanism to protect our CNS system. And well, regarding this, my own experience is, the blood-brain barrier is so important to protect our brain. But there's the other side, that when we have a problem inside the brain parenchyma, inside the brain disease, when we send a drug, they cannot reach the brain. That is a problem of blood-brain barrier. For example, if there's a tumor, I actually observed when you inject some dye into the bloodstream, I can see this dye is coming out where in tumor area, indicating there's a disruption of blood-brain barrier. So that's can be kind of used for studying this part. But I'm not an expert of blood-brain barrier, but this is a very important aspect of studying, and especially drug delivery into the brain or CNS, or central nervous system. So another example is to facilitate drug delivery into the brain. Some people are trying to use a focused ultrasound to disrupt blood-brain barrier temporarily during the time, allowing more drugs can enter into the brain tissue. So very important question.
Thank you very much. So 네, 한국어로도 질문 부탁드리고요. So while other students are typing, I have one more question maybe, if there's time. So the optogenetic stimulation of the parasites was also very fascinating. So you showed that stimulating the parasites can actually so constrict, basically stop the blood flow.
Yes.
But so I was thinking that stopping, I'm not sure if there's a lot of applications for stopping blood flow, but there would be a lot of treatments that require starting the blood flow, right? So
yeah.
Can we stimulate the parasites in a different way so that they can actually work to open closed blood vessels?
Yeah, very important question. So that this one was out of curiosity, but actually this field of parasite, the role of parasite, whether the parasite constrict the blood, especially capillary, has been very important for people who are considering strategies for stroke therapy. So the big issue in stroke area is, so you may have heard that there is within stroke, within 3 hours, you have to go to the hospital, ER right away, so that you may get injection of thrombolytic drug. which opens up the blocked blood vessel. However, that's not the end. Actually, even after this opening of the big arteries, which was blocked, even after this, more than 60% of patients still suffer from the downstream constriction of the capillaries. And that people believe to be involved by the parasites. And so the constriction mechanism of parasite is somewhat considered as very important to understand the mechanism to boost the current limitation of blockage of a blood vessel opening treatment. So hopefully maybe this can be helpful to your question. And there's another question by Soyun. How could you define the vector in blood flow? Yeah, thank you for the question. So the vector is a quantity that is defined as direction and amplitude. So in blood flow, you actually raises a good question. In this case, I think what we have shown as the velocity vector is actually based on the speckle velocity vector. And there's another way to prove that what we measure is actually relevant to, let's say, blood cells, which is inside the blood flow. So we did the experiment to show that velocity versus white light imaging or fast imaging to show RBC by manual tracking. Those velocity vector and the one we measured are actually comparable. That's what we had to perform for the paper like revision process. So thank you for a very important point.
Thank you for the nice questions. Any more questions, comments? So maybe there's time for one more question from me. So related to the last question, so in the speckle image velocimetry, so basically the speckles have to come from the single vessel in the plane, right? If there's other vessels underneath, they're gonna interfere in the measurement. So was there a way to make the speckles only come from the specific plane of vessels that you're interested in?
Yeah, thank you for that question. I think this is a kind of a critical question. The one I showed is those speckles are representing the whole blood vessel. But even the blood vessel we are looking at from the top is a kind of a surface blood vessel, and that represents as a 2D space. What I think what we believe, what we measured is kind of representative, a change from the speckle. So if the blood vessels are very thick, then obviously we know that the blood vessels are like this thick. There is a kind of more complicated three-dimensional distribution. So we may not be able to reveal all this aspect. So that's why I consider the current technique has a limitation onto about 2D representation. I think there's much more work to be done to make this one even further. And also, like, we are also curious about how to extend this technique into the 3D aspects. Mm-hmm. Thank you for comments.
Mm-hmm. Yeah, I think that it's a new technique, so I think it's very, very interesting and it has bright future. Thank you very much. 또 질문 없으신가요? 학생분들.
네, 시간 뭐 1분 남았으니까 한글로 질문 주시면 네. 그것도 괜찮겠고요.
마지막 질문, 마지막 기회입니다. 네, 부끄러워하실 필요 없고요. 예, 영경 학생 하나 더 질문.
하고 싶은데,
예.
페리사이트에 뭘 조절해가지고 그 뱃살을 꽉 쥐게 만드는 거예요?
Okay, the question by Yeongyang was she's curious what mechanism actually can squeeze the parasites, what I actually showed you. I think this is a very critical and important question because there have been a big debate in the field of this. For example, in Neuron Journal in 2015, there's 2 papers actually telling parasite is not responsible for capillary blood flow because it doesn't contain any actin, smooth muscle actin inside. while the other paper actually shows the indication of, hey, parasite is be able to constrict the capillary based on the experimental evidence. So this is actually even currently a debatable aspect, but the definition of parasite is a little loose because from the main vessel until the capillary itself, there's a different composition and types of parasite. Some of them contain smooth muscle actin. So regarding to Yeongyung's question, I think this is a critical question that the one I showed is it's early infancy that we haven't been able to get down to the mechanism study yet. So there's a lot more work to be done, in fact, afterwards. And yeah, you have asked a real critical question, and thank you for that.
감사합니다.
네.
So any more questions? Okay, if not, I think, yeah, for the interest of time, we should stop here. And thank you so much for the very, very interesting talk. And yeah, looking forward to having you here soon in person.
Yeah, I'm so sorry. I wish I can actually meet face to face with these many interesting students. Hopefully next time. And you are welcome to ask me any questions by email even afterwards. So hopefully see you guys in person again soon. Thank you.
감사합니다.
수고 많으셨습니다.